Let's explore how CRISPR-Cas9 recognizes the correct DNA target with incredible precision. CRISPR-Cas9 is a molecular machine that cuts DNA at precise locations. But how does it find the right spot among billions of base pairs? CRISPR uses 3 distinct recognition layers to ensure accuracy. The first layer is the PAM sequence, which acts as an initial filter scanning the DNA. PAM is a short sequence, typically NGG, that Cas9 must find before it can bind. This dramatically reduces the search space. PAM is NGG, where N can be any nucleotide base, and it appears roughly every 8 base pairs in the genome. The second layer is the seed region, which requires perfect base pairing. The seed region, typically 10 to 12 bases adjacent to PAM, requires perfect Watson-Crick base pairing. Even one mismatch here prevents binding. The third layer involves the full 20 nucleotide guide RNA sequence verification. All 20 bases must match sufficiently for Cas9 to proceed with cutting, though some mismatches far from PAM are tolerated. Let's see how these layers work together during the actual binding process. Step 1. Cas9 scans along the DNA, searching for PAM sequences. This is rapid and nonspecific. Step 2. Upon finding PAM, Cas9 tests the seed region. The guide RNA probes the DNA for perfect complementarity. Step 3. If seed matches perfectly, the DNA double helix unwinds, forming an R-loop structure where guide RNA hybridizes with target DNA. Now let's understand why sequences that are almost identical still get rejected. A single mismatch in the seed region creates structural distortion that prevents R-loop formation, causing Cas9 to dissociate immediately. The binding energy of a perfect match is much greater than a mismatch, making the interaction thermodynamically unfavorable. Mismatches far from the PAM are sometimes tolerated because R-loop is already stable, but they reduce cutting efficiency significantly. Cas9 uses conformational changes as an additional proofreading mechanism. Cas9 undergoes conformational changes from inactive to checking to active state. Only perfect matches trigger the active cutting conformation. The catalytic domains only align properly when the complete recognition sequence is bound, providing a final checkpoint before cutting. Let's look at the numbers that demonstrate CRISPR's remarkable specificity. For perfect matches, the dissociation constant is around 1 nanomolar, indicating very tight binding. With just one mismatch in the seed region, binding affinity drops over 100-fold, making binding much weaker. This graph shows how cutting efficiency drops dramatically with each additional mismatch, especially in the seed region. Let's summarize the multi-layered recognition system that ensures CRISPR accuracy. The recognition hierarchy filters potential targets through multiple checkpoints: PAM scanning, seed matching, full guide verification, and conformational proofreading. Each recognition layer independently reduces the probability of off-target cutting by 100 to 1,000-fold. Combined, these layers achieve specificity ratios between 1 trillion and 1 quadrillion to 1. This sophisticated multi-step verification system is what makes CRISPR-Cas9 a precise and reliable tool for genome editing.